Till last video we done with this morula and blaustula structure. Today we'll go with the events of gastrulation. But before I proceed with that one, there are few topics which you need to know about like this much. So I would love to introduce again these all things. So let's start with the morula. Morula is simply when 60 cells combine. So what you are thinking, what 60 cells? So first thing, here's the ovum and sperm. When they both get fertilized, they form a structure which is also called as fertilized egg or zygote. These all happen in the fallopian tube like fertilization. Then they simply copy themselves and they divide into other existing cells which are called as blastomere. After that, they'll form another structure which is about which is having 4 cells. That does not mean here then comes the main part. This structure called as what? That structure called as morula, which is simply when 60 cells combine. Morula, basically that was raised, that's why I've mentioned again morula, which is simply 60 cells combined. They are quite loosely packed as other cells, and they form a structure which is called as berry shape. So in last video we discussed this all and the blastocyst formation. So if we just take one cell, we'll see zona pellucida, which is simply the outer covering, then blastocyst formation. Blastocyst hatching is simply when they lay egg. Hatching simply means laying egg. So when we talk about hatching, this means we are considering birds into that. Then comes the main part which is about the diagram. So I could make it again. So let's see, here comes the main part. Here the outer, you'll see a few other cells. If you just take this one cell, that's called as cyst basically, but when you take the entire outer, that's called as trophoblast, right? This trophoblast is again divided into two parts, which okay, let's mention here. Okay, so here come these both part, which is called as tutorial and the placenta. This placenta, you might be thinking this for providing nutrients and that all, but placenta simply helps in protection. When comes about this one, that's called, they simply provide nutrition. Well, then in this round structure, the cells come over here. There's these cells called as inner cell mass, or even we can just consider them as embryoblast. You'll see written in this paragraph which we discussed in last video, or even I'll show you later on. Then comes one more part. It's like cistern, then trophoblast, then inner cell mass, and the cavity. That cavity is having another name. The name of that cavity is called as blastocele. And even this have another name which is mentioned in our book. So here's that name, which is also called as segmentation cavity or blastocele. So here you can see the diagram. Here's the blastocele cavity, trophoblast, and these inner cell mass. So I'm sure it will be clear, hopefully. Then comes our today's topic, which I mentioned. It will be about events of gastrulation. So let's start with this one. Okay, before I proceed with this one, there are few points I need to write it, so I'll back with that. So before writing it, I would love to make it with the diagram. So as mentioned, zygote will be formed, then blastomere, then 4 blastomere, then morula, which all things were done, then trophoblast. So I'm just directly making another structure. So after this structure, this simply structure is like here are the trophoblast, the inner cell mass, the cavity called as blastocele, everything we discussed a few minutes back. Then after this structure, you'll see one more structure which will be like this one, which will have an outer cells and inner like this structure, and the middle one like this. So what are these? These are simply formation of germ layers. How they'll form: the outer one you see is called as ectoderm, the inner one you see, not this one but this inner one called as endoderm, and then this middle one called as mesoderm. So these three are the germ layer which you'll discuss, right? I'm sure it will be clear. Then their outer, like here, let's make with another color. Here you'll see one more structure on these edges. This structure is simply called as villi. These are basically chronic villi. So chronic villi, or when we just call them as a uterus tissue, because uterus tissue, basically these are present on the uterus tissue. So what will they do? When we just combine these both structure, combine in a sense that when we consider them as these both, So they are simply connecting with the blastocyst, which will simply in the uterus. So they'll try to connect with the lining of the uterus so that they they're connecting themselves. Then mesoderm, yes, this mesoderm layer, which will basically arrive later on, as we have three germ layers. The first one, ecto, will arise earlier. The outer one, endo, which is also the innermost, which will also arrive, but the mesoderm comes a little late. So I'm sure it will be clear. Then in this structure I need to erase this all. Hopefully it will be clear. Then let's see, let's go with orange color now. So here comes one more structure like this one. This is structure called as placenta, which is about nutrition to fetus, providing nutrition to fetus. And even this has so many functions. This help in exhaling and inhaling while the child is in the womb or the uterus. So exhale and inhale also done by this placenta. Their hormone release, hormone like hcg and hp hcg and hpl, these both hormone also secreted by this one. So what these hormones are, I could introduce them. So hcg, which is simply human chronic gonadotrophin hormone. Then come out the HPL, that's simply human placenta lactogen. So lactogen you might have seen on few milk powders. That's are simply lactogen. You might have seen a powder which provided to the child when they are born. That's why there's written lactogen. This means they are going to feed up them. So I'm sure it would be clear. So I hope it will be clear. Now let's see what's written in the book so that we understand it nicely. So let's start with events of gastrulation. First thing you need to remember, this is a quite important stage because all things happen throughout these complex cellular movements, resulting the formation of three germ layers, which are ecto, endo, and the mesoderm. So these are a vital stage in embryonic development, and these layers will develop into various organ tissues such as lining of the digestive tract, the heart, the nerves. And yes, basically these three germ layers have quite different function. They first one carry out different things, second one carry out different things, which I'll discuss in a flow chart way so that you understand it more nicely. So here comes the process which begin with the formation of primitive streak, which is followed by the invasion, which is epiblast towards the midline. So we'll see that, as well as the endoderm forms, which is the third layer, which means the inner one. Then the mesoderm, which develop between the endoderm, and the ecto is the first one, which is the outer one. So as discussed about the structure, you can see here the trophoblast, which is the outer cells as mentioned here. Then these cells are called as inner cell mass. Here we have ectoderm. So yes, here you can see something, these three layers like structure. Hopefully I'm not going to shade it because it's looking so bad then. Well, okay, let's take yellow color now. So here comes this cavity, which is called as amniotic cavity, and which has ectoderm. So before I consider this amniotic cavity, which I'll need to introduce. Okay, so I'm just making it over here. So the trophoblast, which is converting to two more layers like this one, you'll see kind of structure. I'll just directly make this kind of structure. So here this the uterus, first one, uterus, and here is the placenta, right? Then comes the main part, which have three coverings like this one. Here 1, 2, and the last one 3, right? So let's see, here we have fetus and everything inside, right? This fetus and everything. Then this inner one cavity, which is having fluid, that's fluid called as amniotic fluid, and which simply protect fetus, which is present inside. Then comes which also helps in absorbing protein shocks, and these all which you understand in the later videos, protein shocks. Well, when we discuss about these coverings, we have three coverings as I have drawn. So let's see the inner one, which is called as amniotic layer, amnion layer. the middle one you see called as cranial layer. Cranial layer. The first outer one you see called as elentation layer. So I'm sure it will be clear. Then we have another structure in this one. Okay, the inner structure over here, this is basically called as umbilical cord. This also have another name, but I could not recall it for now. Well, here this uterus, which make line between placenta and uterus. And yes, when these three layers are combined, they form an structure which is called as amniotic. Okay, amniotic, not amniotic. Amniotic sac, which have so many functions, which cover fetus first thing, which contain amniotic fluid, right? So we're discussing about this one, amniotic, as we got this term, so I'm supposed to introduce. I'm sure it will be clear. So I'm sure it will be clear. Then let's read more. Here comes the formation of embryonic development, which is endo, then the later on arrived the mesoderm. So during gastration, inner cell mass, which is hypoblast cells, or the line with the blastocele cavity, resulting in the primitive yolk sac and the bilayered membrane called as epiblast. So epiblast is simply kind of a structure you'll see on the 8th day after the fertilization. So if I just make it round like a structure over here. Okay, I'm sure it will be clear. Okay, so here if I just make this structure, you will see. Then this have a outer covering like here. Okay, it will look mess like so, which have an outer covering, not covering, but the fluid like structure. This fluid like structure is called as synchronotrophoblast. This is simply part of trophoblast, which is signed. I'm just writing trophoblast something. Then the inner one here, which we get the cell, this called as cytotrophoblast, you can write it. Then this cytotrophoblast, they form a structure over here. So this structure have two layers. These two layers are simply epiblast and hypoblast. Like this is two layers which are epiblast and hypoblast. These could be shown on the 8th day after the fertilization, so I'm sure it will be clear. Then comes our epiblast, which is split into these amniotic endoderm and the embryonic epiblast, which mentioned. Then embryonic epiblast develops into the embryo, including ecto, endo, and meso, and the germ cell. Germ cells are simply, we just consider these all layers as the germ layers, but when they combine, that are called as germ cells. While the extraembryonic forms yolk sac, which is simply the egg middle one part. The trophoblast develop into several stages, eventually becoming chorion, which mentioned, and economically dried portion of the placenta. So trophoblast, as I mentioned, they are divided into two more parts, chorion something. Then the another, which is placenta. So they provide nutrition and do such functions. Trophoblast induce the mother's urinary cells to form the maternal portion of placenta, which is deciduous, which is a structure which is become rich in the blood vessels, which will provide oxygen and nutrient to that embryo, right? Then comes the fat of the germ layers. So okay, I'll introduce in the next video. I'm sure it will be clear. Thank you for watching. Bye bye.